Hi guys, in this topic we're going to be discussing membrane transport and facilitated diffusion. In our previous lecture, we had spoken about selective permeability. This is one of the characteristics of the cell membrane. So you should be able to define selective permeability, and you should be able to explain the features of the cell membrane that allows for it to be selectively permeable. So just a quick review. So selective permeability speaks to the ability of the cell membrane to allow some substances to cross while other substances cannot cross. Now, what are some substances that can easily get across the cell membrane versus substances that do not cross the cell membrane that easily, and therefore they require some amount of assistance or they require energy in crossing the cell membrane? So we're going to start off with the things that can easily cross the cell membrane. So, cell membranes permeability or selective permeability allows for some substances to cross while others cannot cross. Some substances, all it requires is for the substance to be small enough to diffuse through the pores, and also for the substance to be non-polar. So it doesn't have a charge, and therefore it easily crosses the lipid those fatty acid tails in the center of the plasma membrane. And if it's small enough, it can also cross. So anything that is small to diffuse through the pores, anything that is nonpolar hydrophobic molecules can easily cross. And here are some examples of those type of molecules that you need to know. So this is not an exclusive list. However, these are just a few things that you might see mentioned in questions. So hydrocarbons can easily cross. Also, carbon dioxide and oxygen and nitrogen gas can also easily cross these cell membrane because they're small enough and they're nonpolar. All right, here are some substances that have difficulty crossing the cell membrane. So anything that is too large or to charge, and by that we mean ions. So if it has a positive charge or a negative charge, it has difficulty crossing. And also molecules that are polar. So molecules that are has a slightly positive charge and a slightly negative charge also has difficulties crossing the cell membrane. So for example, water does not cross the cell membrane easily, and therefore water requires specific channels like aquaporins that allow for the passage of water across the cell membrane. Um, Here are some examples of molecules that have difficulty crossing the cell membrane. So sugars like glucose, for example, and I mentioned before water. Glucose is too large, and therefore glucose is not easily diffused through the pores of the cell membrane, and glucose needs help crossing the cell membrane. All right, here's a topic that should be somewhat familiar to you guys. So you probably covered this in biology or pre-AP biology, and these are the types of transport. So when we talk about transport, we're talking about the movement of materials across the cell membrane. So how do materials get into the cell? How do materials get out? And that process is called transport. Now there are two main types of transport. Transport can be described as passive or active transport. And the main difference between these two types is pretty much is energy required or not? So the use of energy, and also the direction in which particles are crossing as it relates to the concentration gradient. So we're going to start off with passive transport, and then we're going to get into the different types of active transport. So let's start off with our passive transport. Passive transport is a type of cell transport in which no energy is required to move molecules or to move particles. So particles, they will move in a specific way. They're always going to be moving down the concentration gradient, or they're going to be moving with the concentration gradient. So these are two terms that are used to describe how the particles move. So down the concentration gradient or with the concentration gradient. This suggests that particles are always going to be moving from an area where there is a lot of the particles, so the high concentration, to the area where there's a low concentration of the particles. And if that does not ring a bell, or if it doesn't make sense quite, We're going to talk about the concentration gradients a lot, and we're going to talk about the direction in which particles are moving. All right, so let's discuss the types of passive transport. So I'm going to highlight some stuff on here that you guys must know. For passive transport, this type of transport does not require energy. So no energy is needed for these particles to move. Particles are always going to be moving within or with the concentration gradient, or particles are going to be moving down the concentration gradients. And there are three types of passive transport. We have diffusion, we have osmosis, and we have facilitated diffusion. So those are our three types. All right, let's discuss each type of passive transport briefly. So we're going to start off with the diffusion. Now, all types of passive transport are described as diffusion. Um, and diffusion is pretty much the movement of particles without the need of energy from high to low concentration. There is simple diffusion. So when we talk about diffusion, just in general, movement of small particles, we're really talking about simple diffusion. So this is a spontaneous process resulting in a constant motion of particles. And these particles or substances will always move from an area of high to low concentration, and they're always going to be moving down, or another term that we could use is we say that they're moving within the concentration gradient. So down or within the concentration gradient means that it's moving from high to low. So here's an example of what we just discussed. Molecules are diffusing across the cell membrane. So here we have the extracellular fluid, and this is extracellular means fluid outside the cell. And here we have fluid within the cytoplasm. Fluids can cross the cytoplasm from the outside of the cell into the cell, or vice versa, can move from inside the cell to the outside of the cell. For this picture right here, here we have our area, sorry, here we have our area of high concentration. So there are a lot of the particles on the outside, extracellular fluid, and these particles will move spontaneously, naturally, they will move and they will go inside. So they're going that way. And the reason why they're moving that way is because they will always move without energy from an area of high concentration to an area of low concentration. And this is simple diffusion. Simple diffusion is when we have particles that are small enough to permeate or to go through the pores of the cell membrane, moving or crossing. And there are different rates of diffusion depending on the molecules. So some molecules will diffuse faster, some molecules diffuse slower, and also energy, well, not energy, but temperature plays a role in the rate of diffusion. So temperature can also increase the rate at which particles diffuse. And these particles, because we're talking about cells, because that's all our unit 2 is about cells, always remember that we're describing this in the context of the cell membrane. So diffusion occurs across a selectively permeable membrane. Another type of passive transport is osmosis, and osmosis is regarded as a type of diffusion in which water molecules are moving from an area of high concentration to an area of low concentration across a selectively permeable membrane. And remember, whenever we're discussing this in the context of the cell membrane, we always add this at the end. So this movement is occurring where? Across a selectively permeable membrane, because we're talking about a living cell. So whenever water is moving, that is osmosis, and water will always move based on the potential of water to move. So water, where water potential is higher, so where water potential is lower. So it's always going to be moving from an area where there's not as many solutes to an area where there is a lot more solutes, and these purple dots, those are solutes. And if you don't recall what solutes mean, we did discuss this in our live lecture in class. So these are things that you guys have to brush up on if you don't recall what these are. So I would advise that you guys watch a video, ask some questions when you see me again, or you can read your textbook. Another type or third type of passive transport is facilitated diffusion. Now, this is a type of diffusion. However, what makes facilitated diffusion different from regular diffusion is this word right here, facilitated. Facilitate means to help or to assist. Whenever particles are large, too large so that they cannot easily or simply diffuse through the pores of the cell, and they require assistance crossing, we call this type of transport facilitated diffusion. So not to be confused with active transport, so facilitated diffusion does not require energy. So even though the particles are larger, it still requires no energy in order to move these particles. So these are particles that need help crossing the cell membrane. And what helps these particles crossing the cell membrane? Transport proteins. So proteins that are responsible for allowing particles to cross. So these proteins could undergo transformational changes in order for these molecules to cross. Facilitated diffusion increases the rate of diffusion. So this is a faster um, type of diffusion because of that assistance that allows particles to cross. So what are some of these particles that needs to be helped or assist across? We have small ions, water itself, and water is facilitated by a type of transport protein called aquaporins. And then we have carbohydrates that also need help crossing. So for example, glucose needs assistance crossing the cell membrane. There are two categories of transport proteins. So transport proteins can be classified as carrier proteins, or they can be classified as channel proteins. Each transport protein is specific to the substance that it facilitates. So not all substances can be facilitated by any transport protein. So transport proteins are very specific to their the things that they carry. All right, so we're going to talk a little bit more about facilitated diffusion. Facilitated diffusion does not require energy, as I said earlier, and it is indeed a type of passive transport. Oftentimes students will confuse this with a type of active transport, but it's passive. There is no energy required for substances to move because these proteins are helping these substances to move. So channel proteins, so we did say that there are two types of proteins. So we have channel proteins and we have carrier proteins. Channel proteins, they provide, as the name suggests, a channel for molecules and ions to cross. So these channel proteins are typically hydrophilic, and they're also regarded as being gated channels. And as the word gate suggests, these require specific, and these are usually like the ion ones. So these require specific molecules to open the channel or open the gate, or it requires a specific concentration of the molecules in order to open the gate. So the gated channels, it requires like a specific stimulus. So there has to be a specific molecule that comes in order for that gate to open. So it doesn't just open to anything or any particle that requires transporting. An example, a very, very prime example of a channel protein would be aquaporins. And aquaporins are very, very key, guys. And I hope you guys, if you don't have this in your notes, put it in your notes. Try to review or read up some more on aquaporins. Aquaporins are protein channels that are specifically designed for transporting water across the cell membrane. So if you ever wonder, water is one of the most vital compounds to all living things. Cells need water. However, remember the cell has the fatty acid tails in the center, and fatty acid tails do not interact with water. So if water want to get from side A all the way over to or down to side B on the inside of the cell, how does it do this when it has to cross these fatty acids? Well, here are protein channels, and these are gated channels called aquaporins that allow for water to cross without water having to interact with the hydrophobic tails, and also the hydrophobic tails do not have to interact with water as a result of these protein channels. So key thing from this slide here, aquaporins. If you don't know what those are the specific channel proteins. All right, so we did mention two types of transport proteins. So we mentioned the channel proteins that we just discussed, and now we're gonna talk about carrier proteins. So carrier proteins are a type of proteins that serve a role in facilitated diffusion. However, these guys they undergo what are called conformational changes, and I'm just going to highlight this word right here. So they undergo conformational changes. So what they do is they conform, or they're changing their structure in order to transport these substances. And my little diagram right here is depicting a carrier protein in which once the particles are inside, it pretty much changes the shape. So for example, this part is open. Once the molecule is inside, this part will become closed, and the other region right here is now going to become open. So the protein is undergoing conformational changes. But we know a lot more about proteins, right? Because we did cover proteins in Unit 1. So we know that when we talk about proteins changing its shape, this is as a result of a new molecule being added to the protein, and therefore the folding of the protein is now going to be changing. So protein is going to be folded in a way that it allows for specific substances to be carried across the cell membrane. So think of proteins changing their shapes in order to now meet the requirement, or in order to be able to transport whatever molecule it is that needs assistance crossing. All right, I added a few practice questions on here. So these are just very simple questions checking for your understanding. I don't want to make my videos too long, guys, so I'm really working on that. So I'm going to go ahead and I'm going to just have you guys pause this video and you're going to attempt to answer the questions. And if you have any questions about these, I will talk to you in class. And the answers to the questions are also on my lecture slides that I shared with you guys in Canvas. Right, here's another question, and we're going to give this one the same treatment. We're not going to go over it. We probably go over this in class if we haven't already done so. If you have questions about this, please let me know. The answer is also shared in the lecture slide that corresponds with this video. All right, so now we're going to be talking about active transport. So remember earlier we said that transport is the movement of molecules across the cell membrane. And there are two types of transport. There is passive transport, and then there is active transport. Now, passive transport does not require energy, and molecules are always going to be moving from an area of high concentration to an area of low concentration. Now, for active transport, this movement is going to be against the concentration gradient. So it's moving not from high to low, but from low to high. And a key thing with active transport is that this process requires energy. So energy is needed in order for substances to be transported actively. And because energy is given to this process, substances can pretty much move against the concentration gradient. So they don't have to go from high to low. They can actually go from low to high once there's energy provided. Now, what are some examples of this, or what are some mechanisms that are in place along the cell membrane for active transport to occur? So we have pumps that can pretty much pump ions. We have co-transport molecules that can help to transport other molecules by pretty much going through some changes or having a cofactor. And then we have exocytosis and endocytosis. And I'm hoping that you guys, once you hear or see these words, it pretty much jogs your memory, because you should know what these two types are if you don't know what the other types are. And those are the types of active transport. All right, so let's talk a little bit about the types of active transport. Before we go into the type, let's talk about what is it that allows for active transport to occur. So energy is needed. The energy molecule for living things, for every living thing, is ATP. So ATP is adenosine triphosphate, and below I have the chemical structure, or the chemical configuration of the ATP molecule. And this molecule is pretty much responsible for storing energy. So this is our energy source. So once we have ATP, it means that we have stored energy, and that ATP can be used to pretty much release energy when needed and is converted to ADP. So ATP can transfer a terminal phosphate, and that's one of its phosphate because it has three. So it has one, two, three phosphates. So if energy is needed, simply a bond is going to be broken here, and then this phosphate group is removed. And once this phosphate group is removed, energy is released, right? Um, and this pretty much changes the shape of the transport protein to better move substances. So once that occurs, and we're going to get into that a little bit more. And this is also a type of conformational change. So whenever proteins are changing their shape, this is what we call conformation. All right, so now let's talk about another type of active transport. And this type of active transport is achieved by the use of pumps that are also found on various parts of the cell membrane. So these pumps are responsible for transporting ions into or out of the cell. And what these pumps do is they maintain the membrane potential. So these are what the pumps do. They simply maintain membrane potential. Now, what is membrane potential? So I want you guys to try to understand and remember this definition. A membrane potential is an unequal concentration of ions across the membrane. And this is important because it results in electrical charge. So whenever there's an unequal or uneven concentration of ions across the cell membrane, this is a good thing because it results in electrical charge. And this is what pumps do. They maintain this unequal concentration of ions so that an electrical charge is sustained. The cytoplasm is relatively negative in comparison to the extracellular fluid. So inside the cell, the cytoplasm specifically is going to have a more of a negative charge than compared to the fluid or the extracellular fluid or the extracellular surrounding the cell on the outside. And here are some examples of pumps. So you have sodium potassium pumps. And these are, for example, these pumps in general, they're called electrogenic pumps. So these electrogenic pumps, they are proteins that generate voltage across the cell membrane, and which can be used later as energy source for cellular processes. So this can be used during other cellular processes as energy. And here are some examples of electrogenic pumps. So we have the sodium potassium pump, and as the name suggests, sodium potassium, the two ions that are involved in this type of pump are sodium ions and potassium ions. Animal cells, for example, will regulate their relative concentration of sodium ion positive and potassium ions. Um, Three sodium ions will get pumped out of the cell while two potassium ions will get pumped into the cell. So if three sodium ions are getting pumped out, that's more than the two potassium ions that are getting pumped in. And this results in an overall net um, charge of one. So there is a one positive net charge in the extracellular fluid. Because remember, so let's go back here. Three sodium ions are getting pumped out of the cell, which means that outside of the cell, we'll have more ions than on the inside of the cell. So more of these positive ions are going to be on the outside because three gets pumped out, and two of the positive potassium ions will get pumped into the cell. So overall, the outside of the cell is going to be more positively charged. And here's another example of pumps. So we have the proton pumps, and we're going to talk about proton pumps a lot when we talk about photosynthesis and well, primarily photosynthesis and also like cellular respiration because that proton concentration is also important for those two processes. So proton pumps think of proton pumps as hydrogen pumps. So these pumps are used by plants, fungi, and bacteria, and they're responsible for pumping hydrogen ions, so positive hydrogen ions, out of the cell. So these are hydrogens that are now ionized because they have lost their electrons. So they're now positively charged because the hydrogen, as you know, only has one electron, and if you lose the electron, then the entire atom is now positive. So, so when we talk about proton pumps, we're pretty much talking about hydrogen pumps or positive hydrogen pumps. All right, so here is somewhat of a little bit more of a specific example of how sodium potassium pumps work. So here we have this diagram, and let's try to understand the parts of this diagram. So here we have the extracellular fluid, which is on the outside of the cell, and here we have our cytoplasm, which is the inside of the cell. And this is our pump. So this is our sodium potassium pump. And sodium potassium pump is responsible for maintaining that ion potential. So what it pretty much does is it will pump sodium out of the cell, and it pumps potassium inside the cell. So that's pretty much what it does. And here we have sodium is represented in red, and we have our potassium represented with the orange circles. So animal cells have a relatively high concentration of potassium ions inside the cell compared to the outside of the cell. So here we have a high concentration of the potassium ions inside, and then outside in the extracellular fluid, we have a low concentration. And the reverse is so for or sodium ion. So animal cells have a relatively low sodium ion concentration inside the cell compared to outside the cell. And if you guys recall from the previous slides, 3 sodium ions are going to be pumped out of the cell at any given time, and only 2 potassium ions will be pumped in. So outside is going to be slightly more positively charged than the inside of the cell. So whenever the protein or this pump, because it is a protein, has this specific shape, and remember these are just used for the sake of a diagram. We know the relationship between the shape of a protein and its function. So whenever a protein has this specific shape, the protein will have a high affinity for sodium ions. So depending on how that protein is folded, it will determine what are in what direction it's going to be pumping. So in this shape, protein has a high affinity for sodium ion. So whenever it's open on the inside and closed on the outside, the affinity for sodium ions is higher. Um, ATP, which is our adenosine triphosphate, will phosphorylate the protein. And what this big fancy word means is that it's going to be adding a phosphate to the protein. So here we have the proteins, and what ATP will do is, so this is ATP molecules and this is our protein. So ATP will be adding that one phosphate group, and once that one phosphate group is added to the existing protein, it will cause what is called conformational changes in the protein. And once conformational changes or the protein conforms, it changes its shape. And once it changes its shape, it's now going to have a higher affinity for potassium ion as opposed to sodium ion. And that's when it's going to be starting to pump the potassium ion inside of the cell. All right, so this is what I pretty much explained in the previous slide. Once that conformational change occurs as a result of the phosphorylation of the protein pump, so here we have our protein pump, a phosphate group is added, and you see how the protein has changed its shape, so now it's open towards the extracellular fluid as opposed to how it was originally. So this phosphorylation is what causes that conformational change, and once that conformational change occurs, it means that the affinity for a specific ion now changes. So in this new shape, the pump now is more attracted to the potassium ions. And potassium ions are going to be pumped outside of the cell, or potassium ions. Yeah, so potassium ions are now going to be pumped inside the cell, sorry. And this diagram is just showing you how when this pump loses the phosphate group, how it reverts to its original shape. And once it is in its original shape, now it has a higher affinity for the sodium ions again. So in a nutshell, this is just pretty much showing you that this pump right here is a protein that can change its shape depending on whether or not a phosphate group is added to this protein. And once a phosphate group, so any functional group that is added to the protein, causes the protein to change its shape, and if the protein changes its shape, then it's also going to be changing its function. So with sodium, with a phosphate group added, the protein has a higher affinity for potassium, and without that phosphate group, it will have a higher affinity for sodium ions. All right, so let's talk about this last type of active transport, which is co-transport. So co-transport suggests that two molecules are being transported at the same time. However, they're being transported in different directions. So one is going against the concentration gradient and one is going within the concentration gradient. So co-transport is the coupling of a favorable movement of one substance with the unfavorable movement of another substance. So in essence, when we talk about favorable movement, we're talking about one is going to be moving, one of the particles or one of the substance will be moving favorably, which means that it's going to be moving pretty much passively. It's going to be moving from high to low concentration, or it's going to be moving within or down the concentration gradient, while the other substance is moving unfavorably, which is suggesting that that substance is moving against its concentration gradient. And two substances are being moved. However, the direction of movement is going to be different. So favorable movement is downhill diffusion, which is good. So that is passive. And then unfavorable movement is uphill, which is more active. And plants are good examples of co-transport in which plants will move sugars and amino acids in this type of way. For example, sucrose, which is a sugar, and the protons or hydrogen ions are co-transporters, which means that they move at the same time. However, they're moving in different directions. So one is going to be moving favorably, which one is moving from high to low, while the other is moving from low to high. So sucrose travels into the plant cell against the concentration gradient, and this is the active transport part of it. But this happens only if it is coupled with the protons or the hydrogen ions that is diffusing passively um, in passively down its electrochemical gradient. An electrochemical gradient is suggesting that this is an ion, it's being moved as like a electrical impulse. So the key thing to know for co-transport is that two molecules are being moved at the same time. One is moving in the direction of high to low, while the other is moving in the direction of low to high. So only one is being transported actively, while the other is being transported passively. So here's a little just like animation to help you guys to understand how this co-transport works. So here we have sucrose, which is large, and sucrose wants to get across the cell membrane. However, sucrose is now paired with protons, so hydrogen ions. Hydrogen ions are moving within their concentration gradient, so they're moving from an area of high concentration to an area of low concentration. However, this sucrose will be paired with or coupled with the hydrogen. So when hydrogen goes in, or hydrogen ions rather, goes in, hydrogen ions are going from high to low. However, sucrose is going to move in the opposite direction, which is low to high. So just to be clear, just to be crystal clear, guys, they're both gonna either go into the cell or they're both going out of the cell. However, once they get to where they're going, one, which is the hydrogen ion, will be moving passively and it's going to be moving within its concentration gradient or down its concentration gradient. However, the sucrose that is now paired with that ion will be moving against the concentration gradient. So the energy is really needed for this part of it, which is the sucrose, while the hydrogen pretty much is moving passively. So that's what we mean by favorable or unfavorable. And if you guys want to take a time to just pause the video and just like try to make sense of this little cartoon animation to see if you can better understand what co-transport is. All right, so here are some common types of active transport that you guys already know. Whenever large substances are being transported across the cell membrane, and if they're moving from an area of low concentration to an area of high concentration, which means that they're moving against their concentration gradient, we call these cytosis. So there are two types of cytosis. Cytosis could be endo or exo. If the cytosis is endo, it means that the substances are entering the cell. And if the cytosis is exo, it means the substances are exiting the cell, so they're leaving. So exocytosis is a secretion of molecules via vesicles that fuse to the cell membrane. And when they fuse to the cell membrane, they will eventually leave the cell membrane. And they do this by vesicles. So these vesicles, these are membrane bound temporary little organelles that contains substances. They move, they fuse to the cell membrane, and then once they fuse to the cell membrane, they pretty much expel their contents and they become a part of the cell membrane. Um, the exocytosis is when these substances are exiting, so they're leaving the cell. An example of these are nerve cells when they release their neurotransmitters. So whenever neurotransmitters are released, this is typically how they. And then we have endocytosis. So remember, endo means inside the cell. So this is the uptake of molecules from vesicles. So it's moving in the opposite direction from vesicles that are coming from the outside, and they have fused with the cell membrane. And then once they're fused with the cell membrane, they will pretty much um, deposit their contents within the cell. Here are some examples of endocytosis. Phagocytosis is an example of endocytosis. Pinocytosis is also an example of endocytosis. And we have cell-mediated receptors or receptor-mediated transports. So here we have phagocytosis is cell eating. So these are when particles are large and the particles are being engulfed by these lysosomes. And these particles are usually like waste particles or pathogens, and they will be dissolved because lysosomes. And remember what lysosomes do. Lysosomes will digest these enzymes. Sorry, lysosomes will digest these molecules using enzymes. The cells will surround the particles using pseudopoda, and pseudo means false, and poda suggests feet. So they will use the pseudopoda structure, which are like feet-like structures. They're similar to how amoeba moves using their pseudopoda. They will package the particles in food vacuoles, and then those particles will fuse with the lysosomes. And once they fuse with lysosomes, remember lysosomes contain digestive enzymes, so they're going to be digesting or breaking down those particles. Pinocytosis is non-specific uptake of like extracellular fluid. So when you think pinocytosis, and here's like a silly way to remember this, I think pino, like pina colada. And that's a drink. And drinks are fluid or they're liquid. So pinocytosis is an example of endocytosis. However, this involves fluid. So pretty much the same thing as phagocytosis. However, phagocytosis involves solid particles, and pinocytosis includes liquid particles. Also takes place in dissolved molecules, like in protein-coated vesicles, and protein coat helps to mediate the transport between these molecules. All right, so that's just some things that are important to know. All right, so here's the last one, guys. So we have cell-mediated endocytosis, and this is more specific. So this is a specific uptake of molecules with solutes binding to receptors on the plasma membrane. So this is a lot more intentional. This is a lot more specific. It requires a lot more like mechanisms to get this done. So there has to be receptors on the plasma membrane in order to receive these like vesicles in order for the contents of the vesicles to be deposited. All right, so this allows the cell to take up large quantities of specific substances, and when solutes bind to the receptors, they cluster in a coated vesicle to be taken to the cell. All right, and that's pretty much all we have here. There's some questions on this slide. I'm gonna have you guys pause the video, look at the questions if you want to know what the responses to these questions are. I did share them in the PowerPoint or the lecture slides that I shared with you guys in Canvas. I don't want to take too much time, and I think this video is already pretty long. So pause the video, pause the question, and see if you can go over that. If you have any questions, you can always ask me when you see me again during class.